comprised of, in terms of major organs, the kidneys, the ureters that connect the kidneys to the urinary bladder, the bladder itself, and the urethra. The work is going to be done by the kidneys. Now, these are going to be unusual for a number of reasons. First off, if we look at, you're seeing a cross-sectional view of the abdominal cavity from the posterior side here. If you look at the position of the kidney, take note these white lines are representing parts of the mesenteries in the abdominal cavity. And literally, the kidney is outside of the mesenteries. It's against the posterior body wall, behind the mesenteries, not surrounded by the mesenteries. For that reason, we describe the position as being retroperitoneal. It's outside of the mesenteries. Technically then, that puts it outside of the abdominal cavity itself. It's outside of the abdominal viscera. Both kidneys are going to be this way. It's kind of an unusual position for abdominal organs. Now, when we deal then with the urinary system, and I'm going to take and jump ahead just a little bit. Those of you that haven't had lab will do this in more detail in lab. But looking at the frontal section of a kidney, this outer area here is the cortex, the thin outer covering, the capsule. And this region here, what they call the medulla of the kidney. The workhorses of the kidney, the way this works, we've got a tremendous vascular supply going into it. Incredibly highly vascularized organs, and they have an incredible blood supply. Literally, you have something on the order of 20% of the total output of the heart going to the kidneys. That is a phenomenal blood flow for structures no bigger than these are. The reason for this deals with the actual functional units of the kidney. And those, as we see here, are structures that they refer to as nephrons. All right. There are two kinds of nephrons that we see in the human kidney. We have what are called cortical nephrons, because most of the nephron is up in the cortex, the renal cortex. And we have what they call juxtamedullary nephrons, where a fair part of the nephron extends down into the renal medulla. Cortical nephrons numerically are the most common. Juxtamedullary nephrons do the most work because of the way they're arranged. So what we need to do is take and have a look at nephrons. And we're going to talk just a little bit about how they work. Now please keep in mind, in the functional aspects of this, we're skimming a lot. Those of you that have to take physiology later on, when you take phys, you'll spend 2 whole chapters on this, as opposed to what they do in your book in a paragraph or 2. All right, looking at a nephron then. What I have here is a nephron that's unfolded. Literally, if we had a line going right through here, this nephron would ordinarily be folded over this way, so that this was right up against this part here. Components that we have in the nephrons. This large structure here is what they call a renal corpuscle. And then we have this tube, and if you look at this tube, nephron begins here. This tube twists and turns in 3-dimensional space a lot, and is close to the point of origin on the nephron, so it is referred to as the proximal convoluted, because it bends and twists, tubule. Gang, these kidney tubules here, they're what you looked at in histology for simple cuboidal epithelium. You were seeing sections through pieces of these different tubules. Proximal convoluted tubule enters into the descending limb of what they call the nephron loop here. We then go up through the ascending limb into the distal convoluted tubule, another highly twisted tubule. From there into connecting tubules and into a collecting duct. All of the work of producing that which we commonly call urine occurs in this. The bladder and the rest of it is mainly plumbing when you get right down to it. Now, some things I want you to be aware of. First off, if we take a look at this in its proper orientation, both juxtamedullary and cortical nephrons, proximal convoluted tubule here, distal convoluted tubule here, all of this, nephron loop. Old name for the nephron loop is loop of Henle. We see the renal corpuscle here is pressed right up against the distal convoluted tubule. That's important. Has to be there in order for this thing to work properly. What we're going to do is filter some components out of the blood, out of the plasma, and essentially kind of process them as they go through this nephron. So, if we take a look at it in a little more detailed version at the very beginning of it, we have our renal corpuscle there. As I pointed out to the earlier lab, and those of you that were in the early lab know the answer to this, so I did this to you, I'll do this to everybody else as well. First thing, we have a network of capillaries here that's commonly referred to as the glomerulus or the glomerular capillaries. This is a capillary bed. That capillary bed is absolutely unique in the human body. No other capillary bed in the body like this. Because, look at it, based on what you know about capillary beds. Why is this different? Gang, look at it. But that's cheating. You were in class this morning.
Ask a question.
Gang, you've got arterioles on both sides of this. There's no other capillary bed in the human body that works like this. When we've looked at capillaries up to this point, we've either had arterioles going through capillaries connecting to venules, or venules going through capillaries connecting to venules. Here we've got arterioles on both sides. That means it's going to be a high-pressure capillary bed, gang. Now, this glomerular capillary bed lies within what's known as the glomerular capsule. Here again, the old term for this is Bowman's capsule. You guys maybe had this in high school or something. Glomerular capsule describes where and what it is, where Bowman's capsule just describes or names the individual who originally described it. Glomerular capsule has two main components to it. The parietal epithelium, that's what you guys looked at for simple squamous epithelium back in histology. And the visceral epithelium, which are, the cells are also called podocytes. These are these kind of purplish cells with the projections coming off of. Okay, this is going to be a high-pressure capillary bed. As a result, there is virtually no absorption that occurs in this capillary bed. We don't have enough of a pressure drop occurring in it to get absorption to happen here. So the only thing that's gonna happen is filtration. We're going to take and filter some of the blood plasma out into this glomerular capsule. What do the podocytes do? Well, they do a couple of things here. They aid in the filtration process. Filtration is pressure driven. And basically what we're doing is selecting what does and does not go through based on size. So everything from large proteins to cellular components of the blood are going to remain in the blood, while the rest of the plasma with the smaller solute particles in it filters out between those projections on the podocytes. The podocytes also on a secondary level help reinforce the wall of the capillary. Capillary wall is made up of what tissue? In any capillary in the human body?
Simple squamous.
Simple squamous epithelium. It's too thin to support a lot of pressure. So these podocytes also act as kind of a reinforcing mechanism for the capillary wall. The deal here is this. We're going to filter not all of the plasma out, just some of it. And it's going to pass from this capillary bed into the lumen of the glomerular capsule, and from there into the proximal convoluted tubule. It's going to follow a pressure gradient as it moves through the rest of the nephron. We're continually producing or having fluid filtered out here. The rate at which this filtrate, we'll call that fluid filtrate, the rate at which this filtrate is produced is called the glomerular filtration rate. Gang, this is not like cardiovascular system and respiratory system, where rates of things change with different levels of activity. Your glomerular filtration rate remains pretty much constant, whether you're laying in bed asleep or whether you're running a marathon. Since it's pressure driven, we got some issues here, because blood pressure is not constant in anybody. It varies depending upon activity levels and other things, yes? So, when we look at this, we've got to keep glomerular filtration rate absolutely constant, regardless of what goes on with blood pressure. You have built-in mechanisms here that maintain a constant glomerular filtration rate. When you have systolic blood pressures, your systolic pressure reading on your blood pressure can be anywhere from 80 to 180 millimeters of mercury. Anywhere in that range, that's having very low blood pressure or relatively low blood pressure, all the way up to being fairly significantly hypertensive. And your glomerular filtration rate is still going to remain constant. A couple little mechanisms involved here. We won't go into huge detail on, but I do want you to at least be aware of, because they relate to some other things that we're seeing here. First off, this afferent arteriole carrying blood into the glomerular capillary networks, being an arteriole, it has a media component to the wall, yes? It has smooth muscle fibers. This allows it to vasoconstrict at need. If there's a sudden increase in blood pressure, it stretches those smooth muscle fibers, and basically as a response to being stretched, they contract and constrict the vessel, decreasing the amount of blood going into the glomerulus. They call that a myogenic response. Now, one other way we can locally control pressure in the glomerulus is, and again, there are others beyond this. This is just to kind of give you a little bit of an idea. If we look at this, we've got distal convoluted tubule here laying right between the two arterioles. And in this juxtaglomerular complex that they've got labeled out, this group of cells right here in the wall of the distal convoluted tubule, just the side toward the arterioles, are the cells of what they call the macula densa. It's a group of specialized cuboidal epithelial cells that are going to respond to changes in pressure inside the distal convoluted tubule. Okay, if glomerular rate goes up, glomerular filtration rate goes up, we're producing more filtrate per unit time. That's going to increase the amount of filtrate we get in that distal convoluted tubule, yes? Some of those macula densa cells secrete then a chemical messenger that's going to cause those smooth muscle fibers in the afferent arteriole to contract and stay contracted longer, decreasing the amount of flow going into the glomerulus, bringing it back to normal. If glomerular filtration rate drops because systemic pressure goes down below normal, different cells of the macula densa are going to produce another kind of messenger. And that messenger is going to target another cell in this juxtaglomerular complex, here and here. These are specialized smooth muscle fibers that also have a secretory function. All right, what they're going to produce as a result of being stimulated by a chemical messenger from the macula densa cells is a material that we call renin. Okay, in physiology, this is going to initiate what we call a cascade of chemical reactions. In the blood, you have a plasma protein that we call angiotensinogen. Gang, this particular plasma protein is produced by the liver. Here's yet another function of the liver. It produces angiotensinogen and releases it into the blood. You have angiotensinogen in your blood all the time. In that form, it really doesn't do much of anything. But in the presence of renin, renin acts as a catalyst. In the presence of renin, angiotensinogen gets converted into angiotensin 1. Angiotensin 1 is still not going to do anything for us. We need another catalyst here. And the other catalyst is something that they call angiotensin converting enzyme. Describes exactly what it does. It's going to convert angiotensin 1 into its active form. Angiotensin 2. Angiotensin 2 is a chemical that causes, one of the chemicals that causes vasoconstriction. All right, gang. You got to stop and think. Renin is being introduced into the afferent arteriole by those juxtaglomerular cells. All of these conversions, and by the way, the angiotensin converting enzyme is produced by the epithelial cells of the capillaries. All of these conversions, each one takes a finite amount of time for it to happen. This all doesn't happen just instantaneously. It happens very, very quickly, but it's not instantaneous. Yes?
What's produced by the capillaries? Sorry.
That angiotensin converting enzyme.
Oh, okay.
Because we have to have a place where all of these things are coming from, yes? So, renin is introduced into the afferent arteriole and starts this cascade going. Before the cascade is done, we're already well into the glomerular capillaries. Angiotensin II is going to cause contraction of smooth muscle in the walls of the blood vessels. How much smooth muscle is there in a capillary wall? A little bit? Oh, come on, we just covered that when we covered blood vessels and cardiovascular system. That's part of this unit, isn't it? Capillaries only have the intimal layer. There is no smooth muscle. Therefore, it's not going to affect the capillaries directly. So, in this pathway, where's the first place we're going to encounter smooth muscle fibers? Pardon? Which one? Efferent arteriole. That would be the next one in line that would have smooth muscle fibers. So, the angiotensin II causes the efferent arteriole to constrict. Well, the whole problem here is we've got a decrease in blood pressure from this side. If we constrict this arteriole here, that backs pressure up into the glomerulus. It stabilizes the pressure, therefore stabilizing the glomerular filtration rates. If you stop and think about it, this is really a fairly elegant system. There are more components to it than what I've given you here, but it kind of gives you an idea of how this stuff works. Ordinarily, the angiotensin II is not going to be produced in large enough quantities to affect the rest of the body. If you have a sustained drop in blood pressure, you can actually activate enough angiotensin II to cause vasoconstriction to occur in systemic blood vessels outside of the kidney. And therefore it can be involved in helping regulate overall systemic blood pressure. Most of the time, though, you only get enough produced to have to deal with it at the level of the individual glomerulus. So we've got these mechanisms in place, among others, to make sure that our glomerular filtration rate remains constant. Now, you might wonder why we have to have it constant. Well, for that, we need to go back and look at the rest of the nephron. Proximal convoluted tubule here. In the proximal convoluted tubule, you're going to have something on the order of 70% or more of the volume of the filtrate reabsorbed. How's it going to be reabsorbed? Well, those cuboidal epithelial cells have built into their membranes both active and passive transport mechanisms, and they're going to pick solutes up, dissolved materials up out of the filtrate. See, there's some stuff that ends up in the filtrate that we don't want to lose. As an example, glucose. When we're talking about blood sugar levels, we're usually looking at glucose. Now, as filtrate goes through the proximal convoluted tubule, if your blood glucose levels are normal, you're going to have the same concentration of glucose in the filtrate that you do in the blood plasma. And yet, by the time you get through the proximal convoluted tubule, all of that glucose has been reabsorbed by way of active and passive transport mechanisms. You know, we talked about active transport and passive transport back toward the beginning of the semester. Things always, in classes like these, come full circle. A particle utilizing either an active or passive transport mechanism to get through a membrane. Let's first thing the particle has to do? Think simple. What's the first thing the particle has to do in order for any of these transport mechanisms to work?
Come up next to the membrane.
Pardon?
Come up next to the membrane.
Not just come up next to the membrane, but come in contact with that transport mechanism, yes? At the right point. Is that a directed process or is that a random process? Random movement. Okay, here's why we have to have a constant glomerular filtration rate. We have to have a predictable amount of these materials that's going to be reabsorbed into the blood. So, the number of the transport mechanisms on these membranes is set. It's not going to change. The only thing we're going to be able to change then is how much time we have for particles to come in contact randomly with those transport mechanisms. If the filtrate is moving too fast, you don't reabsorb as much as you need to. If it's moving too slow, you might reabsorb more than what you really need to. So to keep it predictable, you have a set filtration rate, therefore a set flow rate through this. They call this unregulated reabsorption. Hey, you know, if we're transporting solute particles out of the filtrate and into the cells and then into the interstitial fluid outside the cells, what happens to the solute concentration in the filtrate as this happens? We're pulling solutes out and putting them into the interstitial fluid. What happens to the solute concentration of the filtrate? Goes down. Goes down. Yeah. May decrease. Oh! That means the filtrate is going to become, because we're moving these solutes out into the interstitial fluid, the filtrate is going to be hypotonic. You remember all this from back in chapter 2, I think it was? Hypotonic with respect to the interstitial fluid. Water then moves by way of osmosis out of the filtrate. Water is reabsorbed passively. So we get a whole lot of water reabsorption occurring in this proximal convoluted tubule as well. All right. Unregulated reabsorption. In other words, everything's reabsorbing materials at its maximum rate. Hey, you know, there's got to be lots of active transport mechanisms working here to accomplish all of this, along with the passive mechanisms. If you stop and think about it, active transport requires the cell to do what?
Use energy.
What?
Use energy.
It's going to use energy. It's going to expend energy in the form of ATP. That means cells are going to have to undergo more respiration. The most efficient way to produce ATP is by way of aerobic respiration. That occurs where?
Mitochondria.
The mitochondria. You would expect these cells to have lots of mitochondria. Mitochondria are the only structures in the human body that use what?
Oxygen.
Oxygen. Why you have to have oxygen. All of this being the case, would you not expect to see then the tissues in the kidney here to kind of be oxygen hogs? They're going to be very, very, very oxygen dependent. Yeah? Now, going back not quite so far in the class, I dearly love the urinary system because it links so many different things together. Going back not so terribly far in the class, if we want to stimulate production of erythrocytes in the red bone marrow. There's a hormone that does that, erythropoietin, produced by certain endocrine cells in the kidney. You see now why? The kidneys are very, very oxygen dependent. If your oxygen carrying capacity in the blood drops, you're not getting as much oxygen in the blood as you need. The way you adjust for that is to increase the amount of hemoglobin, which means you increase the number of erythrocytes. So as they start becoming starved for oxygen, they start secreting more erythropoietin, or some of these cells start secreting more erythropoietin, which results in more red blood cells being produced, more erythrocytes. Let's see now. Kidneys then are a secondary endocrine structure. So far, we've talked about stuff out of the chapter on the cell, chapter 2. We've talked about stuff out of 2 different chapters in the cardiovascular system and endocrine system. And we haven't gotten through the proximal convoluted tubule yet. Yeah? Lots of things get linked together here. From the proximal convoluted tubule, we're going to move into the descending limb of the nephron loop. In the descending limb of the nephron loop, you're not going to have any reabsorption of solutes occurring, but you will have water being reabsorbed. You'll have reabsorption of water, but not any of the solutes. So as we go down through that descending limb of the nephron loop, we're reabsorbing water but not solutes. What do you suppose had happened to the concentration of the filtrates? Goes what? Up. Yeah, filtrate concentration is going to increase all the way down to the bottom of that nephron loop. As we go into the ascending limb, if you notice, the ascending limb has a thin component down near the bottom, and then it all of a sudden gets larger in diameter. When we get into that thick component of the ascending limb, water is no longer going to pass out of or into the filtrates. Anybody remember how water passes through a cell membrane? Pardon? Well, the process is osmosis, but it has to pass through special channels that we called what? Way back in chapter 2. Every day now is going to be a comprehensive exam. Call them, these water channels, aquaporins. The inner part of the cell in this ascending limb of the nephron loop, the membrane facing the, or in contact with the filtrates, has no aquaporins in it, so water can't move into or out of the filtrates. But we do have other active and passive transport mechanisms that allow us to pick up ions, things like calcium, sodium, potassium, chloride ions, things of this sort. And we're going to transport these ions, these solute particles, out of the filtrate and into the interstitial fluid. So as we go up that ascending limb, what's going to happen to the concentration of the filtrates? It's going to go down. We're taking solutes out of it. Yes? All right. This all presupposes that we're maintaining outside of the nephron loop a fairly hypertonic environment. In the ascending limb, pumping ions out helps maintain that. The mechanics of that you can do in your physiology classes. Proximal convoluted tubule, descending and ascending limbs of the nephron loop. All of the reabsorption that occurs there is considered to be unregulated, meaning it just occurs at a set pace. You're not gonna change it. In the distal convoluted tubule and the collecting ducts, there we're gonna have more regulated reabsorption occur. We're gonna be able to change how much of certain things we get reabsorbed or left in the urine. Gang, by the time we get into the distal convoluted tubule and the collecting ducts, for the most part, we have reabsorbed somewhere between 98 and 99% of the volume of the filtrates. So the vast majority of the filtrate's already been reabsorbed here. We're gonna be left with that last 1 or 2% that we can change. Okay. Thinking back to, I don't know, the endocrine system. There was a hormone that affected the kidney. It was produced by the hypothalamus and released by way of the posterior aspect of the pituitary gland. And the name of that hormone was? No? Released by the posterior aspect of the pituitary into the blood. Pardon? That doesn't target the kidney.
Oh.
Oh, I know we talked about this.
Is it vasopressin?
Vasopressin. Vasopressin is also known as anti-diuretic hormone. Hey, gang. In the distal convoluted tubule in particular, we have a similar setup to what we have in the ascending limb of the nephron loop. On the side of the cells toward the filtrate, ordinarily we have very, very few aquaporins presence. What vasopressin does is it causes those cells, they have receptors for that particular hormone, and it causes those cells to incorporate more aquaporins into that membrane. All it's doing is opening up a path for water to take. The environment outside the collecting duct is more hypertonic than the filtrate at this point. So by way of osmosis, once those aquaporins are in place, water moves from the filtrate out into the interstitium. We reabsorb water. Now, there's a whole lot more to this system than what I'm giving you. Actually, this ends up relating to the renin-angiotensin system and a couple of other things as well. But it kind of gives you a little bit of a feel for what's going on here. As vasopressin concentrations in the blood increase, we reabsorb more and more water in the distal convoluted tubule in particular. As vasopressin concentrations decrease, the cell starts reabsorbing those aquaporins, and you get less water moving from the filtrate into the interstitium. You don't reabsorb as much water. If we increase reabsorption of water, our urinary output, the volume of urine that we produce per unit time, decreases. If we reabsorb more water, you get less urine and it's more concentrated. If, on the other hand, we decrease water reabsorption, more water stays in the urine. Urinary output increases. You get more water leaving the body, and the urine is more dilute. The collecting duct is the last place we're going to really modify the composition of the filtrate. Once it leaves the collecting ducts, we consider the material to be urine. Once it leaves the collecting duct, we consider the material to be urine. All right. Now, Looking at more gross anatomical structures. In our typical kidney here. You notice a grain, these renal pyramids? See a bunch of lines in there, yes? Those are mainly collecting ducts. Each renal pyramid here is a mass of collecting ducts that converges on this little projection here that they would call a renal papilla. The filtrate, when it leaves these collecting ducts and enters into this minor calyx, at that point it becomes urine. We're not going to change it anymore. Minor calyces come together to form what they call a major calyx. The major calyces come together to form this region here that we call the renal pelvis. And the renal pelvis then dumps into the ureter. From this point on, all we're doing is having this fluid follow a pressure gradient through the system. And we're just dealing with a series of tubes interconnecting different parts of this system to carry the urine from the kidneys to the urinary bladder. At the urinary bladder, for that I have to jump a bit ahead here. At the urinary bladder, hey, gang, ureters come in, there are two of them, one from each kidney, coming in from the posterior side of the bladder and dumping in at the posterior inferior portion of the bladder, here and here. As the bladder fills, we have down here an internal urethral sphincter. The urethra carries urine out of the body. And then we also have an external urethral sphincter. This kind of arrangement should sound somewhat familiar to you guys by now. These sphincter muscles are masses of circular muscle fibers. The internal sphincter is going to be smooth muscle. The external is going to be skeletal muscle. As the bladder fills and stretches, there are stretch receptors in the wall of the bladder that send impulses into the spinal cord. We have what they call a spinal reflex that's going to occur. And we're going to do two things with this spinal reflex. The internal urethral sphincter stays in a contracted state most of the time. You guys think of another sphincter muscle that we've talked about that's like that? Upper esophageal sphincter, lower esophageal sphincter, pyloric sphincter, yeah? They all work like that. Both the internal and external anal sphincters. Yeah, so there's been several of them that we've talked about that have been like that. What we're going to do is signals coming back out of the spinal cord are going to inhibit that internal sphincter, allow it to relax and open up. And you have another pathway that stimulates, you notice there's a muscular part to the wall of that bladder. And the muscles there are what they call detrusor muscles. We're going to stimulate these detrusor muscles to contract. And what they do is they compress the bladder. As they contract, the bladder gets smaller in diameter. That increases pressure on the urine in the bladder, tending to force it out through the urethra. It's a one-way trip here. The external urethral sphincter is under somatic control. Means it's voluntary. Just like the external anal sphincter, in part of potty training, you learn to control that. So you don't just void the bladder every time it fills. You have some conscious control over when you undergo what they call micturition, urinating. Essentially dumping the urine to the outside of the body. So part of it's reflex and part of it is somatic control. All right? Now, gang, when people think about kidneys and kidney function, mostly they think about the kidneys doing things like, oh, okay, they remove waste products from the blood. That's a little piece of it. We've already seen now that the kidneys are involved in stimulating blood cell production, and that the kidneys can be involved in regulating systemic blood pressure. Just so you know, the kidneys are involved in, oh, I don't know, how about calcium absorption in the digestive system? What vitamin is needed by cells of the intestinal wall in order to absorb calcium?
D.
Vitamin D. We produce vitamin D where?
In the skin.
Hmm?
In the skin?
Yeah, in the integument. The form produced by the integument, though, won't have any effect on the intestine. Once produced by the integument, it enters into the blood. It gets modified in the liver. There's another function of the liver, modifying vitamin D. And that modified form of vitamin D from the liver then has to be modified by the kidney into its final active form that affects the intestine. Oh. So, this adds a few pieces to what we dealt with when we talked about calcium regulation in the blood by way of the endocrine system. Now we also see the kidneys having an effect on the digestive system. Waste products, as you think of them in the blood, are not totally cleaned out every time the blood passes through the kidneys. All you do is reduce the concentration, keep the concentration manageable. The urine produced here is going to tell you a lot about what's going on in the blood. And in fact, one of the non-invasive ways of kind of getting an idea of what's going on in the body is to take a urine sample and do a urinalysis on it. Diagnostics, this is really done quite a bit. Earliest urinalyses were being done back in the late 1800s. Physician would take a urine sample, fresh urine sample from a patient, and basically, they're using fairly primitive mechanisms. One of the earliest urinalyses being done in diagnostics was to detect diabetes. They knew that diabetics had more sugar in their blood than normal. So they would take the urine sample and the physician would do that. And if it tasted sweet, then there was too much sugar in the blood, which was an indicator of diabetes. Physicians don't do that anymore. That's what they have nurses for! No. Nowadays, they've got all kinds of tests, chemical tests that they can do, including just little dipsticks that will detect glucose. Yes?
Sorry, urine, it contains a lot, isn't our, urine supposed to be like saline, basically?
It's supposed to be what?
Saline, like it's clean?
Well, it's sterile,
Well, yeah.
but it's not just saline solution. It has in it a lot of the smaller solutes that we find in the blood.
Mm-hmm.
Some of them, like glucose, should be completely reabsorbed in the nephron. If they're not, that means either the kidney's not functioning properly, or there's too much sugar in the blood for it to reabsorb. So it's a way of determining whether there's a possibility of high blood sugar levels occurring without having to do anything invasive.
Mm-hmm.
So they can do urinalyses and look at the components that they find in the urine and tell a lot about what's going on in the body. It's just one piece of diagnostics that they use dealing with this particular system. Yeah?
How are you able to, how are you able to test positive for pregnancy through urine?
They look for concentrations of hormones.
Like really high?
Yeah. Basically, again, You're dealing with things that would enter into the urine through the filtration process in the glomerulus. Some of these things don't get reabsorbed. So basically, again, you can tell a lot about what's going on in the body by looking at the composition of the urine. If somebody's dehydrated, and if you think about it, if you're dehydrated, you don't produce as much urine, and the urine is darker because it's more concentrated. If you're well hydrated, you produce more urine, and the urine is lighter in color because it's less concentrated. All kinds of things can be figured from this. Ordinarily, urine is pretty much sterile. Short of a urinary tract infection or something of that sort occurring, urine is going to be pretty much sterile. And it's usually going to be slightly acidic in pH. The kidneys are so efficient at what they do that you can live a perfectly normal life with only one of them being functional. Some of you guys have done at least a little bit of stuff out there in the clinical world. Anybody here ever seen a dialysis machine? You have, yeah? How big is it? I mean, are you going to pick it up, put it on your shoulder, and carry it out of the room? A dialysis machine is a very poor substitute for one kidney. It won't do what one kidney will do by itself. So, no bigger than these are, they're incredibly efficient at what they do. Like I say, you're using the kidneys 24 hours a day, 7 days a week. And in terms of how they're filtering materials out of the blood and reabsorbing materials from the blood, that's going at a pretty constant rate, pretty much all the time. All right? So,
How does someone know if one of their kidneys isn't functioning? Like, would
they
it?
might not notice it at all. You'd have to have some diagnostics done. One of the things that they do is, if somebody has had total renal failure, both kidneys become non-functional. One of the kinds of transplant surgery that they've done very effectively is kidney transplants. And if they can't find somebody that's recently passed away that's donating a kidney, then they can take a kidney from a normal, healthy individual, remove one of their kidneys, and transplant it if they're compatible with the new host into the host body. Both individuals end up with one kidney, and they can go out and run and swim and do everything else like any normal person will do, and be fine with just a single functional kidney. Now, if anything happens to that kidney, there's a problem. Something else that's kind of interesting about the way kidneys work in dealing with transplants. It's one of the few organs that you can take an adult kidney and transplant to a small child and have it work. Most other organs, you have to be dealing with someone of comparable, reasonably comparable size, as opposed to a very small child or adult. But the kidneys will actually adjust a little bit in the way they function for the individual. So as organs go, they're really quite adaptable. And as we've seen here, they do more than just produce urine. You stop and think about it, we've got linkages here with endocrine system, with digestive system, with cardiovascular system, and when you take physiology, you'll find link-ups with urinary or with respiratory system as well. Lots of things get tied together in this, what you guys look at in the lab is a fairly simple organ system. All right? Physiologically, it's really fairly complex. Other questions with regard to urinary system?
Okay,
they talk about like when you're out, in like the desert, you're dehydrated, do you think they should or should not drink their pee? I'm sorry, because now I'm like thinking about it, I'm like, okay, so dehydrated is to be super concentrated, but I guess it does have a little water in it then, right? But drinking
They don't recommend you drink it. If you're dying of thirst, they recommend you moisten your mouth with it. But the problem is, because it is concentrated, you're gonna have a lot more sodium and things like that in it. And that's gonna cause more problems than it's gonna solve. Kinda like drinking salt water. Drinking salt water will kill you. Yeah, if you're very dehydrated and you're out trapped on the ocean and you go nuts and start drinking ocean water, it will kill you faster than just not drinking at all.
How is that?
Because it's hypertonic with respect to your blood. It's gonna dehydrate you more rather than less.
Oh, okay.
What about if the salt still remains? I'm gonna say, what if you boil the water and remember the salt content will still remain in the water, which is still correct?
Pardon?
I was about to say, what if you boil the water first and then drink it, but then I was like, the salt content will still remain within the water?
No, the salt wouldn't remain.
It wouldn't? It wouldn't
If you had a way of condensing the water vapor, you would get pure water, but the problem is, people trapped out in a rowboat usually don't have the materials necessary to do that.
If they're so efficient, why do we have two?
If you stop and think about it, there's a lot of body systems that we've looked at that have a lot of redundancy in them. Losing one or even several lymphatic organs does not necessarily compromise your immune system to the point of not working anymore. Because there's so much redundancy built into the system. People that have, either through injury or cancer or something like that, have part of their intestines removed. They become a little bit less efficient depending on how much is removed. You can have several feet of small intestine removed and still be pretty functional. You can have the stomach removed and still be by and large functional.
I guess I think of like the heart, you know?
Yeah, it would be nice if we had more than one.